Okay, so welcome everyone to Keynote Session 2. We have our keynote speaker to kick off this session is Dirk Jan de Koning, DJ. DJ is a wonderful colleague who I've worked with for a number of years, both with the genetics and G3. And as you know, this is a meeting from the Genetic Society of America, and the journals are a big part of that. So DJ has been a great partner at the journals, but also a wonderful representative of quantitative genetics and has some really interesting data and stories to tell us tonight. Welcome, DJ.
Thank you, Laura. Right, thank you very much for the invitation. But before I start my talk, I got a message from the incoming Dean of Carnegie Mellon that someone is having their birthday today. So please join me in a round of applause for our moderator tonight. Yeah, I'm going to talk about the model species I'm working on quite a bit of my time, which is chicken, and in particular laying hens. But before I go and talk about the problem, I want to, of course, acknowledge all the people that have worked on this. There's people from Roslin, where we started all this work. There's colleagues from my current university, SLU. And there is people from industry at Lohmann Tierzucht who stand for about 50% of all the laying hens in the world come from this company. And then there's international and national funding from different places. Just to give you an idea, because we hear about these model organisms and wild populations. Well, here we have agricultural populations, and I just want to give you a feel for how effective selection really has been in these sort of livestock species. So if we talk about broilers, so chickens raised for meat, in the 60s it took about 100 days to get a chicken up to 2 kilos. In 2005, that was down to 40 days. So that's a considerable selection response. Likewise, of course, we want to eat the fillet and not the bones. So in the 60s, it was about 12% of fillet. In 2005, we had it up to about 20. Likewise, the efficiency, and I think that's the real key, is to get a kilogram of growth on a bird. In the 60s, we needed 3 kilograms of feed. Now that's like 1.7, 1.5 kilos. So these are all incredibly quick changes, sort of if you look in sort of terms of selection. Likewise, for laying hens, in the 60s, a laying hen would lay about 230 eggs per year, and we've got that up to more than 300 now. But again, more impressively, in the terms of the efficiency, 1,000 kilograms of feed would give you 5,000 eggs in the 60s, and that's almost doubled to 9,000 now. So when you have this sort of quite, impressive selection intensity on production, of course there is going to be some side effects. And the big one we're talking about tonight is avian osteoporosis, which we've known about for quite a few years. It is a big welfare issue because, after emptying a stable, the barn, there's about 90% of hens that have broken legs or a fracture somewhere, and a lot of these fractures occur before the animals are actually killed. What is the interesting bit, and this is probably why we don't hear a lot about this from the NGOs and the welfare organizations, that it's not specific to any given system. It's in the free-range system, it's as much of a problem as in the cage system. So it's not a unique system to the way we keep these animals. But if you think about, a UK flock of 90 million hens or a US flock of 320 million hens, you can see this is a considerable welfare problem that we need to take serious. To say a little bit about the biology, because chickens have different bones from mammals, because they have hollow bones in which they have a third bone type, which is called medullary bone. And it is this medullary bone that they can lay down to when they're not laying an egg, and when they need to make an eggshell, they can recruit calcium carbonate from the medullary bone into the eggshell gland. So it's meant to circulate between and then replenished by calcium carbonate coming in from the diet. But it's when there is an imbalance that part of the structural bone can also be turned over into eggshell, which is good for the eggshell but not so good for the bone. So already in the 90s, together with this breeding company, they took chickens from a commercial breeding line. So these were the elite birds at the breeding company that were used to select new laying hens. They took a population from that and they started to select those divergently for bone strength for a number of generations at the Roslin Institute. And then in 2000, they took the high line and the low line, and they made a cross between them, as we sort of do in many species for QTL detection. The phenotype, one of the phenotypes is breaking strength, and we use a jig like the one here, with basically 3-point breaking strength, and you simply measure the breaking strength in Newton, and that's the phenotype we use. We can do that on multiple bones and derive an index that way. So the first question we ask when we pick a new trait like this, is there any sort of, is it heritable? And we got quite a good selection response after a number of generations, especially in the high bone index line. The y-axis is, it's a combined score from breaking strength of the tibia and some other bones, but it's basically expressed in Newton. So we did a standard QTL mapping exercise, and we found good evidence for a QTL on chromosome 1. I remind you that the chicken chromosomes, they have huge chromosomes and a couple of medium-sized ones and then microchromosomes. So this is a beast of a chromosome for about 500 centimorgan. So there's a reasonably sharp peak there, but that's more of a scaling effect, so it's a really big region. And this QTL affect mainly the tibial breaking strength, so in the top of the leg. Sorry, bottom of the leg. So then, first thing we did is add more markers to that, and we got a reasonable refinement. We got the area a little bit narrower. But of course, on an F2 cross, you can only get so far. But luckily, this commercial population, this breeding line at the company, that is ongoing. So we can always go back to the founder population where the birds were originally taken from. We just have a later generation. So that's what we did as a next step in 2006. 2,500 birds were sampled from what was then the current generation of the founder population, and we used top and tail for that for the next analysis, which is again tibial breaking strength, but we need to use covariates for egg production and body weight, because they are both correlated quite strongly with bone strength, and we need to make sure we're looking at the correct phenotype. So from these 2,000 birds, we took 950 for genotyping. So here you see the result of that sort of local association study. So on the top is the original QTL graph, and in the bottom you see the corresponding chromosome 1, but now we do an association in the commercial breeding flock, and there is a reasonably strong association under where the QTL peak would be. They're not very big, very significant p-values, but for a local study, I think it's pretty convincing that there is something happening there. So we were quite excited about that we could find the QTL effect from the cross also in the still segregating in the commercial flock. And then what we did was, okay, if we take the best SNP from the commercial line and we fit it as a covariate for the QTL analysis, then we go from the blue line to the red line. So the best marker from the association study explains most of the QTL effect. It's not proof, but it's pretty convincing that we're looking at the same thing. So we're a little bit better there. So then we could make a haplotype with the most significant SNPs. And these SNPs show that in terms of breaking strength, it's a pretty good additive effect of about 30 newtons in breaking strength. You see if you look at the 3 genotype classes, it's pretty additive. So that was good. Now, if we look where does this SNP lie, and this is a pretty poor screen drop from Ensemble, but in the small red circle is the most significant SNP, and then on the right side is the nearest gene, which turned to be RUNX. It's annotated here as RUNX1, but actually we're not quite sure. It's sort of it's annotations as they come for livestock. But actually we were quite excited then because RUNX has lots of functions in development and is sort of central to many things. So, for us, RUNX was an extremely strong candidate gene, so that's what we focused on next. So what happened? As I said, we still have this population at the breeding company, so we went back in 2011 and 2012 to sample more birds, but instead of doing more association studies, we can now start to do functional studies. So, and we took this in sort of 2 steps. In April, we went to the company and we blood sampled birds for genotyping. So we kept them alive, left them in the breeding program, because we can't go in and mess up an ongoing breeding program. That wouldn't be appreciated. So, but we sort of sampled a large number of birds, and then after genotyping, we could identify the birds that were most interesting for our experiment, that had the most interesting genotypes in the QTL region. And so later that year in July, when the birds were going to be killed anyway, we selected those that were most interesting for our experiment to go for gene expression. As I said, because we were most interested, we already had a good candidate, we thought RUNX. So we had only 70 birds, but that was still enough to show a fairly clear difference between the homozygous classes in terms of the bone phenotype, which here is cortical bone density. Because we wanted to isolate RNA, we couldn't do quite the same breaking strength here. And when we look at RUNX expression, we know our candidate gene, there is a difference. I mean, it's not super convincingly significant, but we at least felt that it's still plausible that it is involved in the QTL. But, it's 2012, so like everyone else, we should do RNA sequencing. So, because we had these birds with different haplotypes, we selected 8 birds from each haplotype for RNA sequencing. When we did RNA sequencing, so that's genome-wide, we actually only get 5 genes that are clearly significant at an FDR of smaller than 5%, of which 4 were really clearly in the QTL region. And RUNX, which we also measured earlier by quantitative PCR, it had pretty much the same p-value here, which was 0.09. So there's very good correspondence between the quantitative PCR and the RNA sequencing, but clearly RUNX is not the strongest candidate. So the strongest candidate turned out to be somewhere else. It's over there. So, RUNX is sitting somewhere in that clump at the bottom, and then we have another transcript coming up that is completely off the chart. We could show by RNA, by quantitative PCR, that it has an 8-fold difference there. And so probably by now we were thinking maybe RUNX was actually the red herring, and we should be looking at this other transcript. This other transcript, I'll call it CBS because its real name is unpronounceable to me. But rather than a more developmental gene like RUNX, it's actually very clearly a metabolic gene very much involved in the cysteine and methionine pathways. There is some known diseases for when it's not functional. So yeah, this is now the gene we have been focusing on most. And if you look in KEG, the KEG pathway for cysteine and methionine, it really sits bang on where the red circle is in the middle. Now, colleagues at Roslin have been working further with this because from the RNA-seq data, we could also identify SNPs in CBS, and we found a clear non-synonymous SNPs that is changes the amino acid, but also the polarity of the so it's a different polarity. So at the moment, they're doing functional studies with the different configurations of CBS with the different amino acids. So that is to be continued. In the meantime, our next step was, okay, we found this one effect on chromosome 1, and we drilled in on it, and we have a strong candidate. So what else is there? So we went back to this population that we sampled in 2006, and we genotyped that with the Affymetrix high-density SNP chip. Initially, we submitted 900 birds again for genotyping. We managed to get 750 with good quality genotypes. And again, these are top and tail birds. So as I said, they were typed with the Affymetrix high-density SNP chip that has about 600,000 SNPs. But those 600,000 SNPs, they are chosen across a panel of breeds, so we don't expect all of them to be informative for laying hens. We did the analysis, which was done by an excellent master student, Beati Raymond. He used GenAble for the association study, again using body weight and egg numbers as covariates, and he used a subroutine CG-MISC for post-analysis, which is quite a nice tool that you can link into GenAble. So after the quality control, we had 225,000 informative SNPs. And when I say approximately 750, it's actually 752 that we had for analysis. So here's the Manhattan plot. Again, you see how we have this really big macrochromosomes in chickens, sort of the first 5. And then there is some very strong effects on chromosomes 3 and 8, and possibly also some others more suggestive effects. So that's promising for the future. So when we look at, first of all, look at chromosome 1, because there is the markers in blue here are the SNPs that I showed you earlier from the local association study that we did. And then the yellow ones are all the other SNPs that are on the high-density SNP chip. So it's clear that in the region where we found something, there is some SNPs that are more significant, and then there is also some additional evidence on chromosome 1. So we're now re-genotyping some of those to see where they are or what their effect is. Clearly, there's a much stronger effect that we had on chromosome 3 here, and what this graph shows is like a local Manhattan plot, but all the SNPs are colored in relation to the linkages equilibrium with the best SNP. So the best SNP is sort of the white outline, or the black and white diamond at the peak there, and then all the other SNPs are colored related to the level of LD. And so we can see that all the SNPs around it are in pretty high LD with the best SNP. So that leaves me to think we're probably looking at a single QTL there. Plus we have a plot of the minor allele frequencies underneath. Likewise for chromosome 8, where we also had a very strong, significant effect. What is interesting there is that there is also some additional significant or SNPs that are suggestive, but not quite in full LD with the best SNP. So it'd be interesting to see if there is multiple effects there. Another thing the student did was to take sort of the next crop of SNPs, where we take sort of everything that's more p-value than more significant than 10 to the minus 3, find the ones that are close to genes and throw those genes into a gene enrichment analysis. And then we sort of just have an idea if there is clear processes that come up, and there's hedgehog there, glycine, serine, and threonine metabolism, but none of those are sort of really screamingly significant. But it's at least it's another way of looking at our results. So this was sort of all the stage where the findings are, but what we really want is to improve laying hens, the bone strength in laying hens at farming, under farming conditions. So that was in the follow-up project that's currently going, where the research question really is, these SNPs that we find in these elite lines, do they have any meaning in commercial layers? Because the chickens, the laying hens at the commercial company, they are housed individually or with one other hen, whereas the other ones in commercial farms, they're in bigger groups and they interact much more. So we really want to know whether these SNPs that we picked up, do they have any meaning in real life? So we want to look at the effect of the interactions with the housing regime, diet supplementation, but also whether any of these SNPs also affect the feather score, because that's also something that changes a lot in a laying hen throughout lay. So we did a experiment at our own experimental farm where we bought really commercial layers from 2 different suppliers. So these are the laying hens that are used for production of table eggs, bovines and Lohmann selected leghorns. Then in our farm we have 2 different housing systems. They're either in cages with 8 hens per cage, and those cages are enriched according to EU protocols, or they are in floor pens with about 100 birds where they have roosting sticks. Plus then we have 2 different levels of food supplements, and in total we have more than 3,500 birds in this experiment, and we keep those for 100 weeks of lay, which is also much longer than previously, but this is the new aim, because if the birds can lay longer, then we have lower turnover of laying hens, and that's a big environmental saving as well. So a little bit about body condition scoring, as you may call it, or integument scores. So chickens can lose their feathers or they can be feather packed. And so when they have developed feather scoring systems for chickens, and that's what you see here when you go for a score of 1, which is really not very well covered, to a score of 4. And if you go into a laying hen stable, you see all these different categories. So it is very variable. So at the end of the experiment, we scored these birds several times during the experiment for their feathering scores, and then at the end of the experiment, just to have a trait that we want to test. So then, after the birds were killed, we needed to check that they were still in lay, because once they stop laying, their bones really sort of fill out and gain strength. So we needed to have birds that were still laying. Then we took 1 leg to measure for bone strength and radiography in Uppsala at the Department of Medical Sciences, The other leg was sent to our colleagues in Spain that will do crystallography and chemical composition. Then colleagues from Linköping University, from Dominic Wright groups, they weighed all the combs because they have found an interesting correlation between comb and bones. And then for every 5th bird, we took bone in RNA later, plus another sample in -80. And we took all the livers for DNA. So, by doing this experiment, we're also creating sort of resources for further research and biobanking. So now, at this moment, we're genotyping these birds for candidate SNPs that we picked up in the GWAS and from other studies in bone strength and comb traits. Then we can test these effects in different housing systems and diets, plus pleiotropic effects on these feathering scores. And then we have lots of biobank material for functional studies. So I hope I've been able to give you a flavor of how we can use multiple populations and techniques to identify candidate genes, in this case CBS, as in a non-model species. Also that because of right selection, we can have birds that can lay lots of eggs and have healthy bones. And hopefully that applied science can be both cutting edge and make a difference. Thanks again to all the people that helped me, and thank you very much.
We have time for some questions.
So maybe I missed it. What's the heritability of the trait?
The heritability,
Yeah.
it's about 0.40. This is the heritability of the bone index that was published in around 2000, and I think it was one of the first data sets that was analyzed with ASREML, but that's just by the by. But the heritability is about 0.40, so it's quite strong.
Yeah, I was surprised because the trait is supposed to be probably fitness related, I would imagine. But anyhow.
So just because you didn't see an expression difference in that of runks, you have to is that the only reason that you're kind of moving away from looking at runks? Or do you have, are you really convinced that the other CBS was the initial hit? Because it's kind of interesting when you showed the fuller SNP panel, there was a higher peak. I'm assuming that was near where CBS is.
Yeah, using the... So we'll hopefully be able to answer that question soon, because we, from the sequencing data, we've identified more SNPs, obviously, and we now have a haplotype that better fits the data. So we're going to be validating that haplotype, and that shall hopefully give us more. So that's a haplotype in CBS, and then if that fits the data better, then it's sort of more evidence for CBS. But we're still not, I mean, we don't exclude a role from rungs, but it's very clear that we have this strong expression difference from CBS and no documented evidence in the public domain that rungs and CBS play together in some interesting way. So for the moment, our money is on CBS, but it's very interesting how you can get carried away with a candidate gene. And if it wasn't for the gene expression study, I don't know how long we would have been chasing that one.
So could there be variants that have gone to fixation and that are not segregating in the population anymore that could also cause the phenotype?
Sorry?
So your cross is looking at variants that are still segregating in the population.
Yeah.
Could there be variants that have gone to fixation that are also causing the phenotype that if you outcrossed it, maybe you could find those and make those candidates for things that you could remove to remove the trait?
Yes, that's a good question. We're looking. I mean, the SNPs are all segregating. So and we've been selecting from. We've been sort of taking the selection lines from the founder population, and we've been validating in sort of next generations of the same founder population. For it to be sort of divergent in the F2 cross, it had to be segregating in the founder population. So it seems to be at a reasonable frequency still, which sort of makes us wonder about pleiotropic effects. But of course, bone strength itself hasn't been selected for that hard.
Okay, great. Thank you.
Thank you.